I would like to start this talk congratulating with John. I was considering that nowadays some of your papers are really become classic papers in this field of physics applied to tumors and to mechanics, and this is you're really giving important contribution to this field. This is my subject of my talk. I'm interested in this interaction between mechanics and biology, and with a possible faraway interaction with clinics. What I'm going to say is partially something known, which has been published, but the last part of the talk will be devoted to something new, which is yet unpublished. So what I was speaking about, what is a tumor spheroid? So you plug a tumor cell in a fluid with abundance of nutrient, starts duplicating, and after, let's say, 20 days, you have something which is macroscopic. And so it's a collection, it's a bunch of cells, and it's a good way to address a number of questions of solid tumors in vitro. The idea of modelization of this dates back to the 70s. The early models are due to Greenspan. What is driving the growth, what is driving the size in time is the duplication rate, of course, but also the availability of nutrient, and there are some nice models that I use in teaching, actually, that date back to those days. At later stage, the nutrient which is provided by pure diffusion through the boundaries is not enough anymore, and the spheroid, these multicellular aggregates, start producing something that mimics a vascular network and a new phase person. But in this talk, I'm constraining myself in speaking about which is the early stage. It is 20 days more at most. So you realize to predict what happens, which is the size in time. So if you have some availability of nutrients everywhere, because you still have a small spheroid, you grow exponentially. As a matter of fact, at later times, when you, in the other extreme case, you have that you are large enough that the diffusion occurs only on a on a thin layer at the boundary of your spheroid, so you're undergoing surface growth. So this is a very basic idea of growth of these things. However, they're not completely wrong. If you go to some papers, they provide, of course, this is a cartoon, but they provide some quantitative predictions. But my interest is not much in the free growth of these spheroids, but I'm interested in what happens when you take this assemble of cells and you apply external loads. So what is the interaction with the mechanical stress? This is an idea that dates back to 20 years ago, 30 years ago, the group of Rakesh Jain in Boston. And they did a very, very clever experiment. So they do the same growth in a gel, and then measure displacement of the gel. They know the mechanical properties of the gel, so they know at what extent the gel is providing a load on the boundary of the spheroid. Actually, they see that there is a dependence on the growth versus the stiffness of the gel around. The stiffer is the gel, the more there is an inhibition of growth. So this is a first evidence that there is an inhibition of growth due to mechanical stress. However, this kind of experiment was very nice, but was a little bit difficult to to reproduce mathematically, because the mechanical properties of the gel are not trivial, and then you have a load that applies, which is larger and larger, growing the size of the spheroid. So we go to more recent times, when there was this experiment by a group of Paris Institut Curie à Paris, and Montel and others, and Giovanni Capello, among others. What they do is, the idea is the following. They do not load mechanically the spheroid using some wool, but they applied osmotic pressure. What does it mean? They put a macromolecule in the fluid side. The macromolecule is provides the following. So at the boundary, you must have a jump. You must satisfy a jump on the pressure, which is the chemical potential, which is a function, some function of the density of the macromolecules minus the pressure of the fluid. So discontinuity of which this must be zero, or the tension of the surface, let's say zero. And this makes that the actual role of these large macromolecules, which are at the boundary of the spheroid, is to dehydrate, so that is to load basically, dehydrating the spherical mass, they are loading it. And this is easier. This is an easier example than the previous one, because you control the density of the destron around of the macromolecules. These macromolecules cannot enter, so they out here that cannot go inside because the pores are too small. Now, in this case, you have an exact knowledge of the pressure that you apply, and it's evident that depending on the amount of the pressure, that is 500 pascals and so on, so you have a difference in the growth, which is in the radius, or if you like, the volume of the spheroid. So it's a replication of the result that was already known, but now from a point of view of the controllability, it's more clear. And this is something that we wanted to replicate. So there is something more that we know about the system. The first thing that we know is that there is a pressure inside. So what they do in their experiment is another thing. They measure, let's say, a pressure inside the spheroid, and they see that there is an increasing pressure from, this is the boundary, R over R0 equals 0, and then there is an increasing pressure going to the center of the spheroid. How do they do that? They take some acrylamide spheres, and they look at the diameters of these spheres inside, and depending on the compression of these acrylamide spheres, they go up and find the pressure. So is there any possibility to do a mechanical model which is able to account for what happens here? Which is not trivial, because we should give account both for the growth, which is reduced by the load, and for this non-homogeneous stress inside the material. It is also known what happens to single cells when you apply a hypertonic solution and so on, but it is clear that when you have an aggregate, things are different. The reason is that when you have a single cell and you have to deal with macromolecules outside the cell, the difference is between the diameter of the macromolecules and the pores of the membrane. Here it's different. What you are comparing is the diameter of the destron versus the diameter of the interstitial space between the cells. Okay, the last experiment that we want to face in our theory, and we hope to account for that, is that if you take these spheroids here and you cut them, they open up in not a trivial way, which is schematized here. This is a schema of that. So in some region, they tend to close up, and in other region, they tend to open up. So the external, so from a mechanical point of view, it seems that there is a hoop stress at the boundary, which is open, tends to open, so it's tensile. It is compressive inside. Okay, let's go to the modeling. I write down a usual equation for conservation of mass, balance on forces. I don't know if you are you have some confidence with this, but the basic idea is that the stress depends on the strain versus some configuration. Which configuration? If you have growth in a portion of your material and not growth in some other portion of the material, it's mostly convenient that you compare, you take as a relaxed state, the one which possibly involves that there is not continuity in the material. Material is a reference configuration which is possibly not intact. So it's a long story. I don't go into details. I just tell you that we write down, we include growth, and we include also active tension, because there are active stress in these spheroids, using this what is called multiplicative decomposition of the gradient of deformation. Basically, this means that we are counting into this tensor, which is called capital G, both the after roll of the cells and the possible growth in the cells of the cells, duplication internally that gives rise to residual stress. Okay, this is the methodology. This is a classical representation, and so now our energy will depend on the apparent deformation compared with respect to the reference one. So we choose some strain energy for this material. We don't know very much about which is the correct strain energy for this elastic material, which is very soft, super soft. It's not very much more than, I don't know, a collection of bubbles, basically. And we choose a possible form of the gradient, capital G. Okay, now the machinery is ready. What you have to do is simply to write down the balance of forces, possibly put the boundary conditions, and solve the equation, and check if our prediction are in agreement with the observations. Just let me remark something. This point, I was telling you that what's happening from a mechanical point of view in this aggregate of cells is largely due to the fact that the cells themselves are producing forces. And this is something which is not usual in classical mechanics. I mean, if you take a book, you go to the library here and you say, continuum mechanics book, and you take so large, which was basically taught to build bridges. And you find a lot of information about elasticity for any material. But now we are looking at living materials. Living materials produce forces internally by themselves. And this is what called active stress. There are some possible ways to include the active stress into these materials, which we have chosen one, and we go on into this direction. Okay, this is the idea of photoelasticity. Let me go back. This is what I was telling you before. So I don't see single cells. I see the aggregate. I undergoing homogenization. And what is the role of you see, of capital P. Capital P is a function of the density of macromolecules, and the continuity of the chemical potential tells me that there is a pressure P, which is actually loading the cell aggregate. Okay, so I know it because I know I know pi, so I know P, and so I know the stress that I'm producing on this aggregate. Then I go on, I write down I assume spherical symmetry. I write down the equation, radial stress, hoop stress, T theta theta. I apply the boundary condition. So you see that in the boundary condition, TRR, I put the role of the macromolecules of the Jackson. I take this super simple function of the strain energy. Why I call super simple? Because it's simply quadratic in the strain. It's like a spring, a collection of spring. And this spring cannot have, you cannot have zero density. So I'm penalizing to go to zero density through the law of the TEF. Okay. The simplest possible. I use the decomposition before. I write down, and then at the end of the day, I have an ODE to be solved. I'm not yet told anything about the small g I was introducing before. So there was this small g before, which is reminiscent of the big G that I was writing before, which is on the diagonal of the capital G. Now the question is, what does the growth depend on? This is the key question. And we try to give the simplest answer to this quick question. And the simplest answer is written here. There is a distribution of nutrient that goes from the boundary internally. So this is an exponential decay of C from C naught at the boundary to Z, almost zero, roughly zero, after a length, capital lambda, which depends on the diffusivity. I use this, and I would like to write an equation for G that is able to tell me what's happening inside for the growth. Now, the simplest idea is it does not depend on the stress. It does not depend on the availability of nutrient. There is some simple equation of this type that has equilibrium in alpha. This is too poor because we have seen that we have a dependence on the stress, and we have a stress that does depend on the radial position. So we have reached it a little bit. Now, in this equation, there is an equilibrium now, which depends on the stress, which is written here in trace of S, which is a measure of the stress itself. Okay. Okay, now I know that I go to, in this way, I will go to an equilibrium that depends on the load on the material. But if you solve this equation and there is no possibility, you have no hope to find the inhomogeneity of the stress I was showing before. So the last ingredient I was to include is that the growth depends also on the availability of nutrient, C, which depends on the position. And so at the end of the day, this is the equation. What I've added essentially here, so this is my modeling assumption, but the simplest one. And it is basically that there is an equilibrium which depends linearly both on S, so on the stress, and on the availability of nutrient, which depends on the position. So we run the numerical machinery and we find this solution, which are interesting. That is, we find actually that depending on the load, there are different radius versus these days. So you see days, the volume versus days, which was very similar to the plot I was showing before. And then we have also this dependence of the radial stress versus the position. There is a stress which is growing with the radius, so going with the inner position inside. And this is something that we hope to see because this is something that was measured. And the last result is what is called in mechanics the Hoops stress, which is the tendency of the sphere to open up or to close when you cut it. And this is correct because we have seen that here you see that the hoop stress here in this early portion versus radius is negatives, which negative stress means compression. So it tends to close, which is the same that we had seen in the previous images. But also here is positive, so it opens up. So there are a number of features that are collected in the mechanical behavior of this spheroid that we have been able to reproduce by our model. And this is a paper that was published 3-4 years ago. And we had done also numerical simulation in 3D, which is much more complicated. This is Simone Pezzuta, a past student of mine. Now I would like to use these last few minutes that I can spend to tell you something new. And what we are doing in collaboration with the group of Giovanni Cappello in Grenoble is to understand a little bit more better how does the active stress produced by the cell work. And so the experiment is the following. They take 2 nanopillars. The nanopillars are here, these ones. And between these, they are flexible. So depending if you bend it, and depending on the bending angle, you can recover the force. So they are somehow they measure the forces depending on the bending angle. Then what they do is that they put some cells. They are CT26 cells. They're tumor cells, but they are cells that provide strong force. And they seeded the cells freely here at the beginning, and they produce a tissue, let's say. They mimic a tissue, so they aggregate, they attach to the pillars, and they provide force. So you can measure the force produced by this sample cell, this collective cell, and in this way. Now, this is clear that, so measuring L, or the comparing capital L versus the original length, you understand how much force they are producing. Now, what you want to do is to check. Now, let's go back a moment. What happens when you deliver there some macromolecules raw, and you are so adding to this material, which is already producing forces by themselves, you compress it by some extra pressure, which is due to the presence of depthon. In principle, I'm expecting that L becomes smaller because every time I have any material, which is not a very bizarre material, I add the force, I get a smaller size. Now, what happens actually, which was very surprisingly, I will show you the last slide, but now I first show the model, and at the end I will show the numerical result with experiments, is that surprisingly, when you add the macromolecules, so you load the material, this material relaxes. So the mechanical response to this compression is a mechanical response which can be only of empty nature, which is surprising. That is, the cells tend to produce less force than the one than before. So this is the mathematical structure. It's similar to the previous one. So you see the decomposition. A, B, B are the active strains that are produced by the cells. These are the components of the stress, and this is the equation that comes out with taking for lambda the strains in the longitudinal and transverse direction. But this is very simple and technical. There's nothing to do. Now, what the new ingredient that we are including here, which is needed to understand what's going to happen, is that the force produced by the cell is not independent of the time, but depends on the story. So if you have some knowledge of the materials with memory, this is the mathematical framework of materials with memory. This means that what the cells are going to do as depends on the mechanical or the dynamical on the mechanical history of the cell. So you define this functional f of t, which is something like a measure of the stress in time with an exponential decay. Then you apply just one step load. So the derivative that I was writing there is actually a step. And so the variation is a step. So the derivative is a delta function. I use this approximation. I approximate f in that way. Then I take that the fading memory is much long, is very long, and at the end of the day, I get this equation for my active strain. The change in time in two ways. So there is a recovery, but the recovery is not versus alpha A equal 1, which means I'm not doing anything. Depends on pi, which is the shock that was produced. And this shock was produced, and the new active stress that I produce is reminiscent of the history that I've been going through. Now, this is my last slide, and in this last slide, the experiments. The experiments are shown on the left. So look at the blue line. On the blue line, you see that there is these are the cells that are just attached to the pillars. Here there is the osmotic shock, the red arrow, and there is a small compression which is fully passive. But now, surprisingly, the cells relax and go to, so if you should imagine that, so this was the initial position, and after dextran, there is a small, and then there is a relaxation, which is surprising because they are compressed. And so this is the longitudinal length, which is proportional to the longitudinal stress, and this is the transverse one. Now the question is, but I like a lot working with biophysicists and biologists in general, because they always say, but you are giving explanation for this behavior. You say it's active behavior. Are you sure about that? Let's check it. So they take an inhibitor of myosin, and I think it's chitokalazin, if I'm correct. And they produce the results which are at the right. Now you see the difference here. You see that there is still the same jump, which is the poor material passive jump, which is when you apply the load, then there is a recovery, but the recovery is much smaller. It means that much of the myosin activity has been inhibited, so much of the active force produced by the material is inhibited, and basically there is a very different. Now we are going this work. The idea is to produce more than one step, and to understand if the overall behavior can be understood actually in the terms of the kernel memory I was showing before. And there are it's a challenge. There are very interesting results that I hope we can publish early. And these are some people I'm collaborating with. Luigi Preziosi Torino, many of you know him. Francesco Mollica Ferrara, Davide Ricobelli, past student of mine. Simone Pezzuta, past student of mine. Tatafilo Sinialopoulos in Cyprus, he was early in Boston. Giovanni Garobla, he has this very nice lab in Grenoble. He's doing the experiments. And Pietro Mascheroni, a past students of mine, now work in industry. Thank you for your attention. I'm ready for questions.